Welcome to the Noyo Center Science Talk. It's lovely to see you all. Thanks for joining us on this beautiful day. I'm actually surprised we have as many faces in the room as we do because it's so pretty. It's hard to come in from outside. I'm Sheila Siemens. I am the director of the Noyo Center for Marine Science. And if you don't know our organization well, because I see some new faces, we're a nonprofit in Fort Bragg that is working to improve ocean conditions, ocean conservation, and to educate our community about what's happening in the ocean. We are trying to build a facility out on the headlands. That's our ultimate goal, but in the meantime, we have a little museum in downtown Fort Bragg that we are just about to open. We actually have done a soft opening. We've been closed throughout all of COVID, so we just recently opened. If you're local, please go down and we have a couple of new things to look at. But we took that space largely for two reasons, really, to tell the story of the kelp decline that we have off our coast, but also to have a beautiful place to show our beautifully articulated orca skeleton. And so that has some meaning in our talk tonight. I'd like to introduce our speaker, Jamie Knaub. She's a Ph.D. student in Integrative Biology at Florida Atlantic University. Jamie, were you there when you worked with us?
No, I was still an undergraduate at UNC Wilmington.
Yeah, okay, great. So when we, in 2017, I think it was, when we did the killer whale articulation, Jamie came out as an intern and worked with us on that articulation and did an amazing job creating a bone atlas. Are you going to talk about that at all? Okay, good. Of all the bones, she took beautiful photographs and measurements of all of our bones and really helped us with that articulation and knowing a lot more about our particular skeleton, which is factoring into some of her now graduate work. So we're really excited to have Jamie come back to us, to the family, and tell us what she's been learning from the work she did with us and all the work that she's done since then. So, of course, these talks come to you free, but we really encourage you, if you can, to either make a $10 donation on our website, noyocenter.org, or to join us as a member is even better, a sustaining member. We have, I'll put some things in the chat box that link to those things if you want to take a look at that. But supporting our education is a wonderful thing to do right now. So let me introduce Jamie and turn it over to you.
All right, awesome. Thank you, Sheila. I'm going to go ahead and share my screen.
Oh, Jamie, and I forgot to mention that what we do with the logistics is if anybody in the room has a question for Jamie, just put it in the chat. If it seems appropriate, I will break into her talk if that's all right with you too, Jamie. And
Absolutely.
ask the question. Otherwise, we'll just ask them at the end, and at the end we can all ask. So if you want to try to put in a question during the talk, put it in the chat.
That works perfectly for me.
Great.
Alrighty. Can you enable screen sharing?
Oh, I'm so sorry.
No, it's okay. I know we're all pretty sick of this virtual environment. I know I am.
Yeah, there you go. You should be able to do it now.
All right. Just kind of move some things around. All right, so you all should be able to see my title slide.
Yeah.
Perfect. All right, well, thank you for the introduction, Sheila. My name is Jamie. Like she said, I'm a PhD student at Florida Atlantic University. And today I'm going to be talking about some projects that I was able to help with over the course of my career so far and how they've inspired me in my graduate work. So I'm just going to go over a quick outline. I'm going to tell you a little bit about me, where I'm from and whatnot. And then what I call my cetacean inspiration, which are the orca skeletons that I've been able to work on. The first being in Homer, and I'll explain the half later. And then the second being the Noyo orca. And then I'm going to jump into, I've recently written 2 proposals for projects that I want to pursue during my dissertation. And additionally, I'm going to talk about the current research that I'm doing with shark vertebrae. So if you're not familiar, a cetacean is a whale or a dolphin. So I'm going to use that term a lot. And moving forward, you might hear me say some other terms. If you don't know them, just let me know. But I'm going to try to explain everything that I can. So just a little bit about me. I'm originally from Hershey, Pennsylvania. I fell in love with the ocean and marine mammals when I was about 3, 5 years old. And growing up in Pennsylvania, there's not a lot of activities to pursue that kind of career when you're that young. So as soon as I could, I moved to Wilmington, North Carolina, and I got my bachelor's degree in marine biology. And this was really the grounding for me to get my toes wet in the marine mammal field. I joined the marine mammal stranding program when I was a freshman, and I stayed in it all the way through my graduation. Additionally, I worked in a lab that studied marine mammals, and I was able to use some of the research from the Stranding Network as part of my undergraduate thesis. I had a really unique opportunity while I was an undergraduate student to participate in what's called a study away program, and that's called Semester by the Bay. So I traveled all the way to Homer, Alaska, and this is a really neat program where students can get involved in really hands-on research courses out in the field in a wide variety, but a lot of them marine mammal based. And it was here that I actually got the most experience with cetacean osteology. So I'm going to talk a little bit about that. So here's Alaska, and Homer is right about there. And so when I traveled to Homer, you also get the opportunity to partake in an internship as a part of a Semester by the Bay. So when I was there, I saw that they had a skeletal articulation internship, and that sounded just up my alley. So I reached out to the intern advisor at that point and was like, this sounds perfect for me. It sounds nitty gritty, dirty, and looking at bones, which I hadn't had a lot of experience with that at that point, but I was definitely interested in. And that's how I met Lee Post. And I'm sad he can't be here right now, but I'll hope he can see this later. So if you don't know Lee Post, he helped articulate the Noyo skeleton. He's a self-taught articulator from Alaska. And he really helped me a lot in the support for me to pursue a lot of the things that I'm studying now. And he gave me a lot of opportunities to study different organisms and all types of skeletons. So I wanted to give Lee a little shout out. So you can see in this photo, we're working on a skeleton here. And this is the first project that I got to get involved with. And the first thing that we had to do for this skeleton was excavate it. This was an animal that was found stranded floating in the bay. And after it was towed to shore and it was necropsied, they kept the bones and buried them in a giant box full of horse manure at the time. And it stayed in that box for several years. So then when my year came around and I was there, it was going to be the year to articulate this skeleton. So the first thing we had to do was dig it up. But it wasn't gross at this point. It just honestly smelled a lot like dirt. So we had to uncover all the bones carefully. Here you can see this is, let me get my laser pointer. This is the mandibles here, and then this is the upper part of the rostrum, and you can see all the beautiful teeth. And then this is me holding one of the vertebrae. And we think because of the fact that we tried to, remove the flesh through using horse manure, like letting the microbes break down the flesh and whatnot, that the skeleton adopted this really rich dark color, which is not something you normally see. And something that I think adds to the uniqueness of the skeleton today. But after we excavated the skeleton, we had to go through a washing process to get all the dirt off of it. So this basically was just scrubbing the bones with a brush and getting all the dirt out of the nooks and crannies. I had an internship partner at the time who helped me along this process. And even in this set of series, you can see we're already looking at a large variation in the vertebrae here, just as far as the shape and the structure. And I want you to keep kind of a mental image of all these little nooks and crannies, because I'm going to talk about those later. But what we do after washing the skeleton generally is a whitening process. And this is generally some sort of hydrogen peroxide solution that we let the bones sit in for a certain amount of time. The longer you let them sit, generally the whiter that they'll get. However, with the skull, we had a bit of a hurdle to jump over, because a lot of the bones in the skull are on the inside are really fragile, really delicate. So we didn't want to submerge the whole thing in hydrogen peroxide. So for this, we actually designed a hydrogen peroxide paste that we painted onto the skull, and we let it sit on there for a set amount of time to help whiten it. So following the whitening process, we go to a drying phase. And this is actually more important than what it sounds. You would think that you just let the bones, dry overnight, but it has to be a very slow, gradual process, because a lot of bones will actually crack if they're dried too fast. And if this is a skeleton that you're going to be articulating, it's really important that all the bones are in good shape. So we went through like a series of phases of drying them in open air and then moving them inside. to something temperature controlled and kind of slowly let them air dry. So how the articulation for this animal worked was the class of this cohort for Semester by the Bay was divided into groups, and each group had to articulate a component of this whale. And then we brought all the components together at the end to have a full skeleton. So I was on the vertebrae team, which we started out by matching all the vertebrae so they were all in a line. And then we had to drill these little pilot holes through all of them, because when we articulate them, we generally feed them onto a pole. So it's kind of like beads on a string, if you can imagine. That's how it's articulated. So we start by drilling a pilot hole, and then we have to bend the pole to the shape that you want and drill the bigger hole in the vertebrae, which I would not recommend drilling bone. It's not a very pleasant smell, but we did that. And then you can, like I said, you can feed each of these vertebrae onto this pole in succession. So at the end of it, our whale came together, and it's hanging in one of the buildings for the Kazimak Bay campus, Kenai Peninsula College. And this is just a few of the people that helped with articulation. And this was a really unique project just because Lee has articulated a lot of skeletons, but he had some really unique ideas for this animal specifically, including shaping the outline of the dorsal fin and the back, and the flukes as well. And additionally, a lot of these whales that you'll see hanging in museums and whatnot will have generally a clear epoxy for representing a lot of the cartilage and whatnot. And for this animal, we did a black silicone, just because we wanted to add, you know, orcas have that really cool contrast of the black and the white. So we wanted to add those components to the skeleton too. So if you're ever in Homer, Alaska, you'll have to go check out this whale. You can see even after the whitening process that she retained a lot of those dark, rich colors in the bones, which I think kind of adds to the uniqueness of this animal. So, but with this project, I started a bone atlas. So basically, I wanted a way to digitally represent this animal, kind of like archive it. And using, you know, a basic camera and some black cloth, it's amazing what you can do. I'm not going to show you all the photos today, but I do want to show you a handful just because I think they are really beautiful. This is one of my favorites of the skull. And not a lot of people get to see a whale skull in their life, but you can see there's a lot of like little unique crevices and crannies. And like I said, that color really came through, which to a lot of people, they expect a white, you know, skull, but I think it is very unique. And then here's a picture of the scapula. I just think they're really cool. So now I'm going to talk a little bit about the features of vertebra, because I want to refer to those later on in my talk. And I'm going to use pictures from the Noyo skeleton because I actually didn't take individual pictures of the vertebra from the Homer orca. It was kind of an afterthought, but I do have ones from this animal. So generally your main body of a vertebra is called the centrum. So it's this cylindrical main portion. And then you have two processes that stick out on either side. These are called transverse processes. So you can imagine this is looking at the right side of a vertebra. So the animal's head would be over here and the tail would be on this side. So these stick out on either side of the vertebra. Then we have what's called an epiphyseal plate. This is really important, and I want to cover it a little bit in depth because I'm going to refer to it later. So vertebra are basically a type of long bone in your body, and all of your long bones have growth plates or epiphyseal plates on either end. So there's one on this side and there's one on this side. And they're there to protect that bone while it's growing. However, once it's done growing, that plate will actually fuse onto the centrum to like basically at maturation. And so this plate, you can see in this portion right here is unfused, but it's fused on this side. So I'm going to talk a little bit more about that in depth, but that's basically what an epiphyseal plate is, or I'll refer to it as a growth plate as well. So then we have this projection sticking upwards. This actually will form an arch on the top of the vertebra, and there's a gap through it, which is called the neural canal. You just can't see it in this perspective, but that's basically where your spinal cord, you'll have arteries and veins running through there down the vertebral column. Then we have what's called the articular facet. So this is a projection forward that will actually articulate on the backside of the vertebra in front of it. So that's kind of how they articulate together down the spine. And then we have this like neural or dorsal projection or a spine. So now we're going to take this vertebra and turn it so it's facing you. So now the animal, its head would kind of be like coming out of your computer screen at you. So this is the front of a vertebra, or the anterior part. So again, just to label these over so you can see how they look in this perspective, you have your neural or your dorsal spine. Those are those articular facets. So again, those would articulate on the back side of the vertebra in front of it. There's your neural arch with the neural canal. And then we have our epiphyseal plate. So remember that picture earlier where I showed you the Homer orca when we were washing it? It was all bumpy and rough on the face. So that's a vertebra that does not have a growth plate fused yet. And actually comes apart, and we call them jokingly like cookies, basically, because it's just a round little disc. But an animal where it is fused, the outside of it is generally very smooth. And then we have the transverse processes as well. So in addition to articulating this orca in Homer, my internship allowed me to look at skeletons of a couple other cetacean species, which was really unique. So this is me holding a row of thoracic vertebrae from a harbor porpoise. And you can see those transverse processes here. They're really wide and kind of wider. And that's because that's where the ribs would attach. So that's kind of a portion of the vertebral column where the rib cage would articulate. But comparing that to alongside the orca skeleton, and then I was able to look at a couple other species. This one in particular is a beluga. This really got me thinking about all the variation we see across species. When you think about it, cetaceans generally all use the same swimming style. They just pump their tail up and down to move forward. So how are we seeing this variation? Why is that? Here again, this is another vertebra where it's unfused, and you can see all those little ridges of the centrum. But why is this transverse process this shape and this one's not? So this variation that I got to see hands-on and manipulate in my own hands, this was a really big inspiration for me to kind of pursue why we're seeing this variation across cetacean species. So now I'm going to talk about, I call it Orca 1.5, and that's because this was a really unique opportunity when I was in Homer. There was a orca neonate that stranded that we were able to respond to and necropsy. So I'm going to apologize in advance. This photo is a little grotesque. So if you're not a fan of gross things, you might not like it. It's not too bad though. But we were able to respond to this animal and necropsy it, which was a unique experience for a lot of the students there. And through that necropsy process, we were able to retain some of the bones and go through the same phase I talked about earlier, where you remove the flesh. This animal, instead of burying it, we actually boiled some of the bones to get the flesh off. And then we go through the washing process, and you can see this animal, the bones turned out really, really white. And so this animal was really cool because this is these are some of the epiphyseal plates of that neonate. And this is from the larger orca that we articulated. So seeing those animals side by side when I was there was really cool because I was able to think about, well, what are the variations that we're seeing, across life history of that animal? As it's from when it's born to when it's a full-grown adult, which this animal actually wasn't. It was a subadult. But like seeing those changes of, here you don't even have the neural arches. They're not even attached. They came apart separately. So those weren't even fused yet. So seeing those differences, from a neonate to a subadult really got me thinking about, well, how do those changes happen across time and what's happening with the vertebrae there? So that's kind of what I consider my second piece of inspiration for looking at vertebrae. And the third one is with the Noyo orca. So at this point, I had worked with Lee through 1.5 orca skeletons and a bunch of other projects. And he had called me up one day and said, well, do you want to help with an orca in California? And I was like, that sounds like a dream. Sign me up. And the only pictures I have of myself during this time are I'm always with a camera hunched over some bones, which is pretty accurate portrayal of what I did the whole time I was there. I had a blast. I was only able to be there, unfortunately, for a few days. But what I did was I photographed every single bone individually and then tried to photograph them again as in groups to create another bone atlas. So again, I'm just going to show you a few pictures of that. So you can see this is the skull here. Very different from the brown skull of the other orca. And then here's the scapula as well. And I'll show you, this is a row of vertebrae as well. And so this animal was a big inspiration piece for me, looking at vertebrae along the column. So I want to talk to you a little bit about the regions of the vertebral column in a whale. So here we have our orca and our vertebral column. And similar to you and me, cetaceans will have regions of the column that are the same, but they lack sacral vertebrae because obviously they don't have legs. But they'll have cervical vertebrae, just like you and me, which are your neck vertebrae. Generally in cetacean, these are fused just because they don't have a lot of neck movement. I think specifically in this orca, 1 through 4 were fused, and then 5 and 6 were fused together, and then 7 was separate, if I recall correctly. But generally, they'll all fuse together in one block. There's not a lot of movement there, and there's always 7 of them. And then we have our thoracic vertebrae. So those are generally your rib vertebrae, and they'll have those, you can even see it here. There's some wider transverse processes there, and that's where the ribs will attach. So those will be through your ribs that attach to the sternum. Those are called sternal ribs. And then there's generally a few at the very end where floating ribs will attach. And those are ribs that will attach to the vertebral column, but not to the sternum. So that's why they're called floating. And then the next region is your lumbar vertebrae. So these are getting more towards, past the dorsal fin into the tail. And they overlap a lot with what we call caudal vertebrae. It's really interesting because there's actually a lot of debate in the scientific literature as far where do these two, where does one stop and where does the next one end? It's not really has been clearly defined. So some people say it's when the chevron bones start showing up. And I don't have those pictured here, but the chevron bones lie underneath the vertebral column in the tail, and they allow for muscle attachments basically. But anyway, so you have these caudal vertebrae that'll run through the end of the tail, and they're generally very rounded and they'll get very small all the way through the tip of the fluke. And what we know about growth plates along the column is that historically studies have found that similar to terrestrial animals, cetacean vertebral columns will fuse starting at the ends and then moving inwards. So what that means is you'll start to see the first signs of fusion in the neck region and then in the tail, and then a subsequent shift inward. So you would expect the last fusion to take place around the middle of the column. But now let's take a look at the Noyo orca. So this is a section of, so you can see here, these are where ribs would attach. So this is like kind of late thoracic, moving into early lumbar. And some studies historically will visually inspect the vertebrae and say, this one is fused, this one is not fused. So we're going to do kind of a gradient scale for that. So you can see we're going to use dark colors, and you can see there's hardly a line here. And you can see it's a little faint up there, but these are what we would consider fused plates. They're definitely not going to come off the centrum, and sometimes you can hardly see evidence of where the plate was. And so this lighter color is going to represent a partial fusion. So you can see the line is more distinct. This one even has like a little bit of a gap at the top. That's what we would consider a partial fusion. And then we have something like this, which we would consider unfused. Well, since I physically held this vertebra and I know that the plate wasn't going to come off, you can still see there's a very significant gap. So that's unfused. And a lot of animals, they can just come off altogether, like that younger orca I showed you. So this all makes sense if we're moving from the head inwards, that we see the fusion here first is the most fused, and then we see, subsequently less. But then we move further in, and it gets a little jumbled. It doesn't really follow that pattern, which is what really piqued my interest of what is going on here. So I'm very excited to talk about that myself, Sheila, and my advisor have just submitted an abstract for the Society of Marine Mammalogy. Hopefully 2021 we'll have a poster, but we're basically trying to quantify this fusion. Like I said, historically studies have just physically manipulated the vertebra, and like it's basically on a scale of does it come off, does it not come off? But that's not really a great way for looking into this deeper. Being able to quantify or actually assign some sort of number to this would really help us understand fusion patterns along the vertebral column. So what we're doing is, since I took pictures of every single bone, turns out it worked out in the end run. We're taking those pictures. We can crop them down to just look at the centrum and the growth plates. And so this is, again, oriented so the animal's head is over here, the tail would be over here. So you're looking at the right side of this vertebra. So we crop it, so this would be the face, or sorry, excuse me, the growth plate that's closest to the head. I'm going to call that the anterior plate, and this is going to be the posterior plate. So what we can do is we can convert that image into a binary image. And what this does is photographs are made up of a bunch of little squares that are assigned colors. Those are called pixels. And what binary does is it gets rid of the whole color spectrum and only keeps black and white. So everything either becomes black or white. So it's going to take all your lighter colored pixels and automatically assign them to white. And it'll take all your darker colored pixels and automatically assign them to black. So you can see already that we can visualize that gap with the growth plate right there. So what we do to analyze this then is we actually have to invert the photo. So that makes the air space white. And then we can use a region of interest and a specialized software that will actually count the number of pixels that are white in that region of interest. So specifically, this one had 945. And you can see the difference from, the posterior plate, this posterior region of interest, and the anterior one. You can hardly see a line, maybe a little bit up here, but you can hardly see any gap with that plate. So that number is going to be a lot lower because it has a lot less white pixels there, versus this one, like I said, has 945. And these images are made up of thousands and thousands of pixels. So when you hear 945, it doesn't really mean anything up front until you start comparing them across each other. So this is a really unique way to take something that was kind of historically done, just assigning like a gradient or a fused unfused scale, and actually being able to quantify this. So I don't have all the data, but I did want to give you all a peek at what we're seeing. So here I have, it's in line with the column here. So this is a caudal vertebrae, this is a lumbar vertebrae, and then this is a thoracic vertebrae. And I've assigned a region of interest for each of the plates. So there's an anterior and a posterior plate for each one. And then I ran this analysis. So what we're seeing here is that your posterior plate has more white pixels. So that means it's less fused than the anterior plate. And you can see that here, it's actually the reverse. There's that big white line where that plate is not fused. And so it has a higher number than the posterior plate. And then again, here with our thoracic vertebrae, a very distinct gap for that plate. And it has 21,000 pixels versus the anterior, which is only a little bit over 1,500. So this is really unique because historically what we've thought is that fusion happens at the ends and moves inward. But now what we're seeing is that might not always be true. So stay tuned. Hopefully we get the abstract accepted and I can publish that work. So I just kind of wanted to tie those three stories back together really quick. So again, the orca that I got to work on in Homer, the neonate that also stranded there, and then the Noyo orca on top of it were kind of my three big puzzle pieces for why I want to study vertebrae, just because of the variation that we're seeing, and we're not really sure why. So looking across species, why one vertebrae is different than another, or looking across life history, why does it fuse at this time in its life, or why doesn't it? And then also along the vertebral columns, why is this section fused when this one's not? So this is where I'll tie in what I'm hoping to do in the future with cetacean vertebrae. So I'm going to talk about two proposals that I've written. I've not started these projects yet, but it's kind of what has been inspired to do based off these three animals. So the first was a proposal to NSF, and I specifically am looking at the epiphyseal plates, or those growth plates, fusing to the centrum. So here's our vertebral body again. And this image is basically just going to kind of turn it on its side and give you a cut view into it. So vertebrae are made of what's called trabecular bone. So it's very spongy bone is what you could relate it to. There's lots of little gaps and ridges and whatnot. And here you can see it showing those end plates again. So historically, what people have done to investigate this bone in cetacean vertebra is they will scan them using a CT scanner. So what that is basically it's like a CAT scan that you get at a doctor, and you're able to look into it. So you're actually able to like image what the inside would look like. So historically, people have looked at that, and when you scan it, it looks kind of like this when you digitally represent it. You can see a lot of those little holes and gaps, just like a sponge. So what studies have done previously is they've looked at the bone in the middle of the vertebra, which is very unique. There's been a lot of really cool findings on this as far as what deep diver bone looks like versus a shallow diving species. What I'm really interested in is looking at that interface of the vertebral body with the end plate. And to my knowledge, no one has looked at this yet, but I'm hoping to CT scan cetacean vertebrae and look at that interface with the growth plate to look and see what are the differences across species, what's the difference across life history as the animal grows up, basically. Are there any differences in sex? And also along the column. So looking kind of inside that plate, the same way I'm looking at it in the Noyo orca with the fusion state, but actually looking inside and looking at a cellular level. So this is something that I've applied for NSF funding. It was recently rejected, unfortunately, but I'm hoping to be able to continue this project without funding, using a letter of authorization to partner with stranding programs near me and attain vertebrae and scan them. So you'll have to check in a couple of years, and hopefully I'll have some interesting results for that. The next grant that I wrote was for the Marine Mammal Commission. And this was recently submitted. It's still pending, so fingers crossed. But this is an interesting project because it relates to a conservation issue we're having right now.
Can I interrupt before you move on? We had a question that I think is related to what you just got done talking about. So
yeah, absolutely.
Mark is asking, he says, it's great stuff. Do the left and right sides and top and bottom, for that matter, of a vertebrae have relatively the same degree of fusion, which is basically what you're saying is you're seeing really a lot of variation, right?
Yeah, that's a great question. So I don't know anything about dorsal ventral. I've seen in literature that it's been suspected that the anterior plate fuses first. I don't really know like what the backing for that is. But it's just interesting to think about because, you know, we have an animal already that's kind of disproving that. So I really honestly personally believe, again, this is just my insight, I have absolutely no data for this, that I think it really just might depend on the animal. But, you know, with this Noyo orca, maybe that was, you know, just a case where a couple of the vertebrae, the posterior plate fused first. But historically, what I've seen in the literature as the anterior plate will fuse first, or that's what's reported at least. But the dorsal ventral aspect would be interesting to see if the top half or the bottom fuses first. That's a great question.
Great, thanks.
Yeah. So this proposal is written again for the Marine Mammal Commission, and this is really tackling a current conservation issue right now, which is the North Atlantic right whale. The North Atlantic right whale is a large baleen species on the east coast of the United States. It's a very slow moving animal, and it's actually critically endangered right now. I think the last estimate is like there's less than 300. It's probably even less now because I've definitely seen a few deaths this year. But basically this species is very prone to both ship strikes and entanglement, and they're basically the highest rates of death in this animal. They basically will come in contact with some sort of line and a buoy system and a series of traps that are all kind of tied together. And when these animals interact with these lines and these traps, when you think about it, whales can't really like swim backwards. They can't like back out of entanglement. So they're stuck carrying this gear on them for what can be months at a time. And what you have to think about is cetaceans generally are swimming, they're thriving in a very buoyant environment. So when you add something like hundreds of feet of line with hundreds of traps on it, you have to think about all that weight and all those drag forces and what that's doing to the skeleton. These animals swim by undulating their peduncle or their tail. So that's, you know, a wave motion of the vertebral column. And when you suddenly add all this extra drag, all this extra weight, what is that doing to the bone? This is something that has been kind of briefly addressed through necropsies. But again, when you're looking at an animal with your own eye, you can't really look into that bone. So what I'm hoping to do is to actually CT scan vertebrae from animals that have been what we call chronically entangled. So that's basically an animal that's been carrying gear for a long portion of its life. And they'll generally succumb to it through either starvation or exhaustion. But there's a lot of these animals that are their skeletons are already archived because it is so critically endangered. So taking these vertebrae and CT scanning them, which is amazing because it's non-destructive too. So it's something that you can borrow a museum specimen and then give it back. So taking a look inside the bone and seeing what's happening there. Is there anything happening with the trabecular structure on the inside through chronic entanglement? So keep your fingers crossed. I'll be finding out about this funding in the next couple months. So at this point, I'd like to just take some time to talk about my current research. So everything that I've done up to this point and what I hope to do in the future has been cetacean based. But right now I'm actually working on a different organism, which is sharks. But their vertebrae are still very cool. So I jokingly say that sharks have got your back because this research is really unique and it could actually help people in the future. So just like you and me, sharks have a vertebral column that's made up of individual vertebrae, also similar to the cetacean. But what we're seeing here, the big difference is that they're actually made of cartilage. They don't have bone. Their vertebrae is hardened cartilage. And I'm going to show you a look here. We can generally divide the column into kind of two regions, anterior vertebrae and posterior vertebrae. And the anterior vertebrae, you can see they're very cylindrical in shape. This is their basically their neural arch. And then the posterior vertebrae, they differ because they have what's called a hemal arch. So it's basically just another arch, but on the bottom. So their vertebral column is comprised of these anterior vertebrae and posterior vertebrae. And what I'm doing is I'm looking inside of them to see what the structure is like. Because you can see there's like all these little slits in it. And I'll show you some images here in a second that are really cool. So at this point, I've been talking about CT scanning. And what that is, like I said, it's similar to a CAT scan that you get at a doctor. We have what's called a tabletop sky scan, Bruker sky scan. So this is a big bulky machine that you have an X-ray source on one side. And what that does is it emits an X-ray through your specimen. Your specimen is generally like in a tube or on a post, and it will rotate at tiny little increments. And every time it rotates, it'll shoot another X-ray through it. And then on the other side of the machine, there's a detector that will pick up that X-ray. And so as your specimen rotates over a series of hours, and you get thousands and thousands of X-rays, we can use software to take all those X-rays and stitch them together. And then we're able to look inside your specimen like digitally. So I'd like to show you all a video of that now. So this is a shark vertebra that I scanned. And you can see it's got all these little kind of slits along the side. And this is the coolest part, is that you can actually slice into it. And then you can rotate it another way and slice into it in another way. So this is really the beauty of CT scanners. And it's really been groundbreaking for a lot of new research. So what I'm doing is I'm CT scanning shark vertebrae, and I'm looking across two orders of sharks. So it's basically two phylogenetic groups, you can consider it. First being the Carchariniforms. So these are things like blue sharks, spinner sharks, lemon sharks. And their vertebrae are a little bit different than the one that I just showed you. This is kind of a whole view. This is a one slice view, and then I'm going to slice it the other way. So if you would cut it this way, this is what you'd be looking at. And shark vertebrae have this really unique double cone structure. And then when you slice it the other way, you can see there's these large plates or wedges. And this is all, again, mineralized cartilage. And then there's these four gaps. And now I'm going to show you what's called a lamniform shark vertebra. So lamniform sharks are like white sharks, thresher sharks, mako sharks. And so their vertebrae are a little bit different. So this is, again, the same orientations, the same slices, but I'd like you to take a look at those differences. So lamniform sharks have these little ridges or like phases of mineralization, and then they have gaps in between them, whereas the carchariniforms have that big wedge. It's a solid wedge of mineral. So I'm still in my preliminary data with the carchariniform sharks, so I'd like to show you some more in-depth data with the lamniform sharks, which is this right here. So lamniform sharks, they have, again, these ridges or these phases of mineralization, and those are called lamellae. And what we do is once we scan it, we can actually count how many lamellae are there. And sometimes you can see the lamellae will branch at a point, and that's what we call a node when it branches. So we scan these vertebrae, and then we count all the lamellae and nodes. And we look at that in the anterior part of the shark, or closer to the head, and the posterior part of the shark, or closer to the tail. And now I'd like to show you some of those images from 4 species of lamniform sharks. So we're going to take a look at the common thresher, and you'll see 2 vertebrae are going to pop up. One will be on the left side to represent the anterior or the head region, and the other one will be on the right side to represent the tail region. So here's a thresher shark scan. And you can see already that this one looks very different than the one that I already showed you. And I'm going to talk about that variation in a bit. We also looked at white sharks, short fin mako, and porbeagle sharks. So you can see here, we're already looking at a huge variation in the same order of sharks. You know, these are all animals that have come to a certain point come down the same phylogenetic line. And we still see this incredible variation in the mineral structure in the vertebrae. And I would like to add that coming soon is the basking shark. Basking sharks are also lamniform sharks, and they're really unique because they're so large. I have vertebrae, but I have not been able to scan them because they don't fit in my scanner. So stay tuned, because I'm hoping to collaborate with Florida University that will have a larger scanner, but they have the similar structure here where you see the radiating plates. I'm very excited to see that. So I want to show you some data from the images that I just showed you, from counting the lamellae and the nodes. So again, this is the lamellae, or those little ridges that stick out. And we have our thresher, the mako, the white shark, and the porbeagle. And this darker bar in the front represents the anterior vertebra, or the head vertebra, and then this represents the ones from the tail. So you can see from this bar chart that it's a consistent trend that there's more lamellae in the posterior vertebra, or the ones in the tail. And I would like to add that species was significant in this. So that means that the thresher, like species, plays a significant role in how many lamellae will be there for that species, which is really unique. And then we're going to take a look at the nodes as well. And so you can see it's the same layout, but we're looking at the node counts now. And there's the same trend that there's more found in the posterior vertebra than the anterior vertebra. And for this one, not only was species specific, but also the anterior and posterior. So that means basically when you look at an anterior vertebra of that species, that's going to be significant to that region, which is really cool. We did not see that trend in the lamellae counts, which is interesting, but I'm going to have to look a little bit more into that statistically. So the whole reason is, why do we see more lamellae and nodes in the posterior region? So when you think about it, how sharks swim is it's a different type of undulation than cetaceans, so it's more of a side to side. And when you think about it, the vertebrae that are in that tail region are going to be succumbing to a lot more bending forces than the vertebrae in the anterior region. As you saw in that little swimming shark, the front part doesn't bend as much as the tail does. And what we think is that this increased mineralization in the posterior region is aiding in supporting the column to succumb to those bending forces. It's actually helping as far as making it more efficient, and it's stronger, that it can withstand those forces. And this is something that's really important, especially in the 4 species I just showed you, those lamniform sharks, because they're some of the fastest species out there. And the last thing I'd like to talk about is specifically one of those species, the thresher shark. If you don't know anything about thresher sharks, they have a really unique, interesting behavior where they have an extremely long tail that they will bend over their head to stun prey. And this is only seen in thresher sharks. And what we think is happening here is that the reason we're seeing the highest counts of lamellae and nodes in any species is because of this behavior. we think that these mineralized ridges, basically these lamellae and all the nodes, actually support the vertebrae in withstanding those immense bending forces, so it can actually do that behavior. This is something we're hoping to publish by the end of the year, but it's very unique and an interesting case. So the last thing I'd like to finish on is, well, Jamie, this is all really cool, but why should we care? So the significance of this kind of research. So specifically the stuff that I talked about with cetaceans, it's really important moving forward because it adds to our understanding of not only skeletal mechanics, but spatial and temporal impacts of vertebral fusion. And that's something that can be applied to terrestrial mammals or even humans as well. Additionally, if I get funded for that North Atlantic right whale study, I'll be contributing to entanglement research. To my knowledge, no one's looked at the intertrabeculae effects from entanglement in whales. And additionally, that'll help our understanding of loading and trabecular bone. So there's a significant amount of research with trabecular bone and loading forces in humans because a lot of humans have back problems, they have back pain. That's how you get ruptured discs and all types of fractures. So understanding this in a different model organism can help us understand it better in ourselves. Additionally, when we look at the shark studies, it's important because it impacts our knowledge regarding mineralized cartilage. There's not a lot of research on mineralized cartilage. There's lots of research on bone, but mineralized cartilage and the form-function relationship it plays in animals is still relatively unknown. Additionally, it will help our understanding of composite materials because we're looking at vertebrae that are made of mineralized and unmineralized phases of cartilage. So looking at how potentially composite materials can be used for strength. And this ultimately can be used for human joint health as well. You know, when you sometimes joints will actually, the cartilage inside them will mineralize themselves. So understanding it again in sharks can help us better understand it in ourselves. So with that, I have so many people to thank, specifically starting with Semester by the Bay. Mark and Debbie were instrumental in the classes that I took and the research that I did there, and have always supported me, writing countless letters of recommendation. So I owe the world to you too. Additionally, Lee obviously had a very big impact. Unfortunately, he couldn't make it tonight, but with everything I've done skeletal wise, he's played a major role in it. Additionally, obviously the Noyo Center with the Orca project, Sheila have always been a support for me, and looking forward to getting this poster out. And all the people that helped with that articulation. I had so many volunteers helping me in those few days to photograph all the bones. Additionally, at my university, my advisor and my lab members, because they help with some of the scanning parameters and emotional support as well. Dr. Natanson from NOAA helped get some of the shark specimens for us. And additionally, Dr. Meredith, she's on my committee, and she is the director of research at the FAU High School. And she also runs the OWLS Imaging Lab, which is where the CT scanner that I use is based. And additionally, I've gotten a little bit of funding from FAU, and the shark research I'm currently doing is funded by an NSF grant that my PI won recently. So there's plenty more I could thank, but I wanted to end that I have a Wix site. I'm also on LinkedIn and Twitter. And additionally, if you're interested in more of the CT scans or anything that the OWLS Imaging Lab does, you can follow the FAU High School Research Twitter or Instagram. But thank you, and I'd be happy to answer any questions at this point.
Wow, so cool. So great to see all that you have done since we saw you last. You've been busy.
Oh, yes.
Answer one question for me while we let people come in with their questions, but we always were under the assumption, or were told, that the plates fused when the animal got older. But we learned since we saw you that our killer whale was 30 years old from the teeth analysis, from the analysis of the teeth, and he still had quite a few plates that hadn't been fused. So what's our understanding in age and all of this?
Yeah, that's a great question. So obviously, like historically from studies, that's always what it's been. It's, you know, when that bone stops growing, when they reach maturation, it's going to fuse. But especially the Noyo orca was really what got me thinking, you know, like you said, this is an adult animal. Why are we still seeing plates that aren't fused? And I think there could be a few reasons. I have to, as a scientist, I have to consider any type of error, first of all, in any of my studies. So something I've considered is that potentially with this animal, in the drying process, those plates may be warped a little bit. Maybe that's why we're seeing a larger gap in some of them. I don't think that's likely, but it's something I just, as a scientist, I have to consider any type of error. But going back to the age thing, that's why I'm really wanting to look, across life history, because that's been kind of the traditional view. But I'm starting to think it has something to do more with diving behavior and, like stroking rates or environmental conditions. So if you think about it, an animal that's going to be deep diving and gliding, maybe it's not going to need those plates to fuse up quite yet, versus an animal that's, maybe more of a shallow diver, does a lot more maneuverability. Maybe those plates need to be fused first. So I don't have a solid answer for you. That's just kind of some of my speculation. But hopefully in like 5 years' time, when I have all this data, I can answer that question.
You said something in the beginning too that I need to circle back to, where you said that the epiphyseal plates actually protect the bone. I mean, I've never really thought about why are the plates there. So what do you mean by that? I mean, I know that's where their bone is growing. So, and it's softer. So,
yeah, exactly. So it's like you have end plates, or those growth plates are on like any of your long bones. So like your femur, things like that. And it's basically a protective cap for that bone underneath to grow. So it's the same thing in vertebrae. They grow outwards as well. So it's basically kind of a protective barrier for that bone to grow. And then generally that's why we see, like, I think that's why it's traditionally been thought too, that when you're done growing, those plates fuse. It just seems that some of them fuse faster than others. So I'm curious as to if that means that the growing has stopped, or if it's from some other implication. But yeah, the trabeculae are very, very spongy and soft. So that plate is kind of a protection barrier for the growth.
okay, and the shark vertebrae scans are just beautiful. I think we need to make some sort of image out of those. I'm like, we need to make some sort of graphic thing and use your shark's got your back kind of thing.
Yeah, absolutely. Thank you.
Yeah. Does anyone have any other questions? I mean, I want to open it up. Yeah, go ahead, Wendy.
I have a question, and that is, well, actually, I was going to ask the question about age, but Sheila beat me to it. But I was wondering, like, especially within the sharks, how much variation is there within a species? Like from one Thresher shark to another. And
yeah,
why?
That's a great question. So it's pretty crazy, but there's a lot of variation. So that's why generally for that study, I'm taking several specimens of the same species and doing scans on. So I think the Thresher Shark, unfortunately, is my lowest count. I only have 2 individuals. But for instance, like the poor beagle and the white shark, I generally have like 6 to 8 specimens, which doesn't sound like a lot, but when you're dealing with these large, predator species that are out in the ocean that generally you don't see carcasses, that's a large study population. But we do see a significant amount of variation across species. And it's crazy in sharks because you even see variation as far as the number of vertebra. So for instance, you can have one poor beagle that has 108 vertebra, and then another one will have 112. So they have a really large variation, not only in the mineralization and how many lamellae, but in the number of vertebra too.
So I had a question. Can you hear me?
Yes.
Hi, my name is Paul. And first question, maybe directed toward Noyo Center. I know this is recorded, and I missed the first half. I'm sorry. How do we access recording later? Is there a way to access it?
Yes, Paul, we do. So on our website, you can go under the what we do, and there's a science talk list with all of the recordings. So give us about a week, but you'll see all the talks up there eventually.
Okay, great.
Yeah.
All right, thank you. And then my other question is, sharks have a cartilaginous skeleton. Wondering, how do you prevent decomposition of the vertebrae, or do you freeze them and then put them in the CT scanner?
Yeah, that's a great question. So a lot of our specimens will come from, either strandings or ones that are called as like bycatch. And that's how we got a lot of the ones from NOAA. They're generally sent to them, and then they'll send them to us frozen. So we'll keep them frozen. And then for the CT scanner, your specimen has to absolutely be still. It cannot move at all. Otherwise, you'll have what's called shifting. So the vertebrae we pack in a canister among sponge, so it's immovable. And believe it or not, if you're dealing with a frozen specimen, even the thawing can cause movement inside the specimen. So we generally have to thaw them to scan them, and then we refreeze them, which is not an issue for what I'm doing. But there's some people who are doing mechanical testing on vertebrae, so they'll actually squish them to see at what point do they break. And that's an issue for that study, because the more you thaw and freeze them, the more they're gonna, they're gonna lose their integrity and they're gonna decompose.
So if you don't mind me asking another, I know I've been involved with scanning some whale bones, and we always had limitations on the size because of our CT unit. The basking shark is a huge shark. So what's the limitation of the CT you're using now, and how much bigger is the next CT you're going to use with, I think you said Fordham University?
Yeah, that's a great question as well. So it's a very large concern, because you pay $500,000 for a CT scanner and then your specimen doesn't fit. So it is a big concern. The vertebrae that I'm dealing with, generally I don't have an issue, but yeah, specifically this basking shark, the vertebra is like pretty much the size of my head. And the limitations on the scanner I'm using are probably about half that. So the scanner I'm hoping to use with University of Florida can take, I think it's like up to double the size of the vertebra that I'm using. So you just really have to be in contact with the right facility. So like I said, Florida does scanning for collaborators. You generally just have to, pay for the staff time to do it, which is why funding is really important, because it's expensive. And then also University of Texas they have several scanners that they do some larger specimens. And there's ways to get around that too. Like you can scan half of the specimen and then scan the other half, but then you run into issues with like pairing them together and whatnot. But yeah, that's a great question. It's always a limitation, is the scanner.
Interesting, just a note. I've been trying to scan some large specimens from whales, and I know out in Utah, the military has a CT unit where they scan missiles. They scan missiles that have a 5-foot diameter. It's hard to get in to get them to scan these bones, but blue whale vertebrae, right whale vertebrae, long dorsal process, it's hard to get it in that space. And I've been trying to convince the military to let me scan some of my specimens there. But on the east side of our country, I'm wondering if there might be some military installations like in Pensacola or somewhere in South Carolina or Georgia that you might be able to tap into also for future research, just so you know.
Yeah, I'll have to look into that. Not that I know of off the top of my head. I think closest to me, Texas has the biggest scanner. And it's kind of like it's not even like an enclosed one. It's basically like the specimen just sits on a post, and I think the scanner actually moves around it. So it's kind of like a big open area, but yeah, I'll have to look in and see. That's a great idea. I've never thought of that. Thank you.
Really interesting research. Thank you.
Thank you.
Hey, Jamie, this is Mark. I really enjoyed your talk. A couple of things came to mind with regards to your point about age. Classic version of cetaceans is they take a long time to physically mature. They grow incrementally, they sort of take a long time to get to like an asymptote. But one of the things that would be fun to look at might be to compare two similar apples to apples, two baleen whales, like a gray whale versus a bowhead whale, an extremely long-lived animal, to see if that fusion coincides, slow fusion, coincides with their great age that they can attain. So I think that would be kind of fun to dig into. And then there was also your point about like ecology or what the species does. Like you could look at, have you thought about looking at beaked whales and sperm whales versus belugas or things that don't dive as deep or as long to address that question, like our Steineger's beaked whales in Alaska?
Yeah, those are great questions. Addressing your first one regarding like a long-lived animal. So there's one of the studies that I looked at in the literature, I think it was from 2015. They did kind of what I was talking about, that historical perspective, the gradient, where they just kind of assess it on a visual or like a physical manipulation scale. They looked at a bowhead. And I can't remember the age of that animal, but if I recall correctly, I'll have to find the paper. There were still some that were not completely fused, which just kind of blows my mind. And additionally, for your second point, it was my life goal. If I could scan a beaked whale vertebra, that would be amazing. I've definitely thought about, comparing some deep divers. There's a student she's now a postdoc at Texas. She worked in my lab before me, and she CT scanned cetacean vertebra, but she looked at what I was talking about, that middle portion, not at the plates. And she found differences among diving behavior, which is really cool. And I've seen other studies who have looked at, 2 or 3 species that have found that as well. But I really would love specifically to look at the plate in regards to those ecologies, just because, that's that interaction there, I think, is really important, more so than the inside of the vertebra.
Yeah, I think the age question is amazing. Bowheads, now people are basically estimating them to exceed 200 years. And then, like, back to sharks, Greenland sharks don't even get mature till 400 or 500. So
exactly. I would love to see, like a Greenland shark vertebra scan would be so cool to look at the mineralization there too. That's what I'm really interested about, this basking shark as well, just because from my understanding, they're large and longer lived as well than other ones. So
yeah, cool.
So, Jamie, did you say you were interested in beak whale vertebrae?
absolutely.
Does it matter if the bones have been processed and dried? Do they have to be fresh with the tissue on it still?
So from my knowledge, for CT scanning, it doesn't generally have a limit for the specimen age or the how it was preserved. It would be more of like for testing strength or something like that. That would be a concern but from my knowledge, no, the freshness does not impact the scan.
Well, the reason I say is I work with the North Carolina Cetacean Center up in Beaufort, North Carolina, and we have a number of beak whale specimens, skeletons, and the group has been assembling some of them. Recently did a Gervais beak whale and a Blainesville beak whale, but we have some that aren't articulated, and I can see the possibility of some collaboration. If you're interested, I'll be happy to put you in touch with the folks at the Cetacean Center in North Carolina, and maybe you might work out something where you could borrow or scan some of those and then return them, the bones back to them after you've done your work. I think that would be an excellent collaboration.
Yeah, I love that idea. Do you work with Keith Ripmaster?
Yeah.
I know Keith. He's awesome. I unfortunately at this point I have all my paperwork submitted because if the people here who have worked on whales know, it's all permitting. So I've submitted all the paperwork for my IACUC and the LOA through NOAA, but everything's kind of at a standstill paperwork wise right now. But yeah, as far as, I'm hoping near future to have that approved and be able to handle the bone and have it legally. So a collaboration to look at beaked whale vertebra would be amazing.
Well, there's a resource for you through Keith Ripmaster in this North Carolina Station Center.
Awesome. Thank you.
Well, okay, we're at our 7 o'clock time. It was so great to have you join us, Jamie. Thanks so much for all the hard work you're doing and for including Noyo and all of the storytelling. Yeah, excellent job. We will get this up on the web within a week. We also send it out on our newsletter, so just sign up for that if you want to hear more about what's going on with Noyo. We are signing up summer camps right now. It's one of our big things. And as I said earlier, come visit us downtown and see what's new. And that goes for you too, Jamie. You need to get out here and visit us again.
I know.
Well, fully articulated. So
yeah, I never got to see it when it was finished, so I really got to make a trip back there for sure.
You're getting a lot of thanks and excellence in the comments.
Yeah, thank you so much for having me. I really appreciate it.
Yeah, good.